For week 3, case study 3, I'm going to be discussing carbidopa and the effects it has on Parkinson's disease. So to begin, I'm going to be talking about Parkinson's disease. It's a nervous system disorder that affects the ability to control movement. It is a disease that begins slowly and then progressively worsens over time. Some symptoms that are found in patients with Parkinson's disease include tremor, slow movement, or rigid or stiff muscles. So what causes Parkinson's disease? It originates within the neurons of the brain. The neurons produce dopamine, which is a neurotransmitter that helps with the communication between cells of the brain. So when the neurons of the brain become either impaired or die, neurons then produce less dopamine. There is not a reason why the neurons become either damaged or impaired, but they do affect the motor movements of the body. Another part of the brain that is reliant on dopamine is the basal ganglia, and that is a part of the brain that is responsible for organizing the brain's commands for body movement. And so the loss of dopamine results in the movement symptoms that are found within Parkinson's disease, such as the rigid or stiff muscles. Another neurotransmitter that is lost is norepinephrine, and that is needed for proper function of the sympathetic nervous system. So because there is less dopamine due to the damaged neurons, it affects the communication within the cells of the brain, as well as the command, the control center, really command system for body movement. Carbidopa is a drug that was established to treat the symptoms found within Parkinson's disease. It's used with another drug, levodopa. And so carbidopa is a decarboxylase inhibitor. It inhibits the aromatic amino acid decarboxylase enzyme and the conversion of levodopa to dopamine peripherally. So dopamine cannot enter the brain due to being damaged to the damaged or destroyed neurons. So a way in which the two act together, so levodopa in the system gets converted to dopamine in the bloodstream. However, because of the damaged or destroyed neurons, it sees the dopamine and blocks it from entering the brain. So the carbidopa works with levodopa to allow it to be converted in the bloodstream, and then it allows some of the the dopamine to enter into the brain. So the X-ray crystallography is a technique used to determine the structure of proteins and the biological and macromolecules. Its goal is to obtain a three-dimensional molecular structure from a crystal. So the crystal structure of a ligand-free dopa decarboxylase was viewed. And so because dopa decarboxylase converts dopa to dopamine, because the L-dopa is converted to dopamine, it's administered with a DCC inhibitor. And the observation of a ligand-free DCC and how it relates to the carbidopa was viewed. So researchers were able to observe the ligand-free DCC and the DCC carbidopa complex. Features of the DCC that were present were the structures of the protein, the cofactor pyridoxal 5' phosphate, which is anchored to the enzyme, how the inhibitor binds, and which amino acids may be involved in the activity of the DCC. And it suggested ways to improve the binding of the inhibitor of the DCC. And then I have the structure of the carbidopa from the Chimera program, and then my references.